Hello, we are Group A, Team 2. In our group, we have Kate DeWitt, Eden Carson, Theresa Laxton, and Derek Wong. Today we'll be discussing cestodes and presenting the Lightowlers study on them. This paper discusses the importance and relevance of vaccinating against cestode parasites, or tapeworms. Cestode parasites cause large production losses in sheep, cattle, and pigs, and can also cause diseases in humans. On the upper right is Taenia saginata specimen, and below that is the disease caused by Taenia solium, neurocysticercosis, in which an infection occurs in the brain. Therefore, research on vaccines for these parasites are quite important. In the Taenidae family of cestodes, there are two hosts in a predator and prey relationship. The predator, such as dogs, foxes, and cats, are the definitive hosts, while the intermediate hosts are usually animals such as sheep, goats, and cattle. Humans are an exception to this and can be either the definitive or intermediate host depending on the species of cestode. Adult tapeworms infect the definitive host, while larvae infect the intermediate host. In the development of vaccines, the aim would be to target one life stage in one of the two hosts. While the definitive host would be a good target for vaccines as there are fewer animals in this category, there is little evidence of immunity development against adult tapeworms. Vaccine development for the intermediate host is effective, however, as past research has indicated that the host is able to mount an immune response against the larval stages of the tapeworms, although the adult tapeworms do survive. Additionally, immunity can be transferred from the antibodies in the colostrum to juvenile animals, providing a high level of immunity for both sheep and cattle. This paper further discusses these vaccines and the challenges associated with the development and commercialization of them.
There are many species of tapeworm. The most prevalent species which Lightowlers discusses in his studies are Taenia solium, Taenia saginata, Taenia ovis, and Anacoccus granulosus. In the case of T. solium and T. saginata, the definitive host is generally a human. However, humans can also be an intermediate host if they consume eggs. The general intermediate hosts for these species are pigs and cattle, respectively. Both species infect the musculature of the intermediate host. Pigs and cattle become infected by ingesting infected vegetation containing eggs or proglatids. Humans may become infected by ingesting the raw or undercooked meat of infected livestock. Eggs or proglatids are then passed through the feces of the human and spread to the environment, where they are ingested by livestock, continuing the cycle of infection. The final species from the Taeniid family I'll talk about is T. ovis. The definitive host is usually working dogs, but can also be wild dogs and urban dogs that come into contact with eggs. The intermediate hosts are sheep and goats, and it also infects the musculature. Dogs become infected when they consume raw or untreated offal infected with cysts. Infected dogs pass eggs through their feces onto pasture. Sheep and goats ingest the eggs with vegetation. The final species I will introduce is E. granulosus. It has an indirect sylvatic life cycle, meaning the pathogen cycles between wild animals and vectors. Humans are usually incidental or dead-end hosts, infected by a vector. The definitive hosts are usually carnivorous predator animals, working dogs, foxes, and dingoes. The intermediate hosts are cattle, sheep, and wildlife. Unlike Taenia tapeworms, E. granulosus infects various organs, specifically the liver and the lungs. Domestic dogs and predators eat offal which contain fertile cysts. Infected species then contaminate vegetation which is ingested by cattle, sheep, and wildlife. Humans become infected if they eat infected meat. T. saginata and T. solium are distributed worldwide. T. saginata is prevalent in countries that ingest raw or undercooked beef, such as Asia, Eastern Europe, and Latin America. T. solium is more prevalent in poorer countries with poor sanitation, that live in close proximity with animals. Latin America, Sub-Saharan Africa, India, and Asia have higher rates of illness due to T. solium. T. ovis is prevalent in sheep-raising countries, such as Canada, Australia, and New Zealand. E. granulosus is also distributed worldwide and is prevalent in sheep and cattle-raising countries, including South America, parts of Europe, and Australia.
Lightowler's paper includes a table summarizing results from several other studies regarding the use of native and recombinant T. ovis oncosphere antigens for the vaccination of sheep. The recombinant antigens referred to are the 45W, 16.17, and 18K antigens. Both antigen types have been shown to induce strong levels of protection above 90% under experimental conditions. These protective immune responses have the benefit of being passed from mother to offspring in colostrum. However, native antigen displays some advantages over recombinant antigens. Native vaccination was able to provide up to 100% protection against field-acquired infections, whilst the recombinant vaccines provided only up to 85% protection. A single dose of the native vaccine afforded a full year of protection, whilst the recombinant vaccine requires 2 doses to be administered every 6 months. Additionally, a second protective antigen is required to allow immunization of lambs with the recombinant vaccine due to interference of the maternal antibody. Based on this information, it would be ideal to vaccinate against T. ovis using only native antigen vaccines, but recombinant techniques must be employed in order to make the vaccine available in commercial quantities. Regardless, both vaccination types have significant positive benefits and are effective tools against T. ovis infection.
The adaptive immune response is important in defending against invading molecules. The response contains many different cells that work in sync to target and destroy pathogens. Parasites release excretory-secretory proteins into the gut, where antigen-presenting cells take the foreign substance and presents the antigen to T helper 2 cells. This triggers a cascade of events that lead to the production of IgE and IgG antibodies. These parasite-specific antibodies attach to the mast cells that are embedded in the intestinal tissue, and they can also neutralize some products excreted by the parasites. The further release of proteins into the intestine leads to the degranulation of mast cells, edema, and inflammation. This is a type 1 hypersensitivity response that has evolved to target parasitic infections. It makes the intestine less appealing to the parasite, which can cause the parasite to detach from the intestine tissue. Cytokines then trigger the release of mucus that coats the parasite, making it easier to exit the body. Granule mediators can also cause damage to the parasite's outer membrane. The adaptive response is an important component in the immune system. It enables the individual to destroy toxic molecules that would otherwise cause illness and possible death. It is able to identify between self and non-self molecules. This prevents the immune system from attacking the individual's own cells. Specificity and memory of T and B cells is a key attribute, as they are able to identify the same antigens excreted from pathogens in reinfections, which enables a larger and more efficient response to the pathogen. Vaccines work by exposing the immune system with an altered form of the pathogen that triggers an immune response. Secondary exposure to the pathogen will cause a faster and larger adaptive response that can prevent a loss of life and money. The T. ovis 45W vaccine in sheep uses non-living antigen from the oncosphere. Recombinant DNA techniques on the 45W antigen were used to develop the vaccine. The immune response to the vaccine triggers the release of specific antibodies that act against the parasite. Alterations to the 45W vaccine with the use of oil adjuvants and recombinant ovine IL-18 increases the antibody levels in the immune response. The variability in the 45W gene may impact the effectiveness of the vaccine due to the parasite differing in their susceptibility to the immune response from the infected host. There have been developments with vaccines through recombinant DNA techniques based from the T. ovis vaccine. EG95 vaccine has been made against E. granulosis and T-sol18 vaccine against T. solium. These vaccines stimulate the adaptive immune response for protection against the particular parasites in future infections.
Despite excellent levels of protection afforded by the T. ovis vaccine, it is yet to be released for commercial use. This is because the economic loss is seen in reduction of the prices that are paid for sheep across the market, which is difficult to quantify and isn't something an individual farmer can rectify by vaccinating his flock. Often infection is not evident until after slaughter. Without the parasite causing direct and significant economic loss to the farmers, there is no motivation to invest in a vaccine against it. This may also be the case with a variety of other vaccinations against Echinococcus and tenuis species. Farmers may be more inclined to invest in vaccination if purchasers were willing to pay more for vaccinated, parasite-free animals. In some instances, particularly if the parasite is of public health significance, government-implemented control programs can fund vaccines and enable their commercial success. In order for a government to be willing to invest in a vaccination program, the vaccine must be of practical value. Important factors include the duration and level of protection, the ability to protect against different strains, and the ability to protect young animals via maternal antibodies passed on in colostrum.
Future prospects described by the paper would include the development of vaccines against species such as T. saginata and T. solium, as previous studies have shown that the larval antigens in these species provide a high level of immunity from infection. Both species can be transmitted to humans, causing disease. Future research may also investigate new methods of vaccine delivery, such as using live recombinant bacterial or viral vectors. Previous research indicates that larval antigens are effective at providing immunity. This knowledge, along with the T. ovis 45W vaccine outlined in this paper, is a good model for the development of future vaccines. This paper suggested the development of vaccines against T. solium infection, as it would protect pigs and prevent the transmission of the tapeworm to humans, thereby preventing neurocysticercosis, which is the leading cause of adult-acquired epilepsy. As of 2016, a vaccine was successfully produced and licensed for T. solium called Sysvax. The development of the vaccine uses the T. ovis 45W vaccine model and technology developed by Lightowlers. Development of vaccines against T. saginata would be useful in the beef meat industry, protecting cattle from infection and lowering production losses from meat infected with cysticercosis. As with T. solium, this would prevent transmission to humans. Following the Model 45W vaccine, there are successful vaccines developed, although it has not been commercially adopted. The prevalence of tapeworms is especially high in developing countries, and so vaccines that are developed must be inexpensive, simple to administer, stable, and have long-lasting efficacy.